Morning all, and I'm very sorry I couldn't be there today. So this morning we'll be discussing genomic prediction and machine learning methods for genomic prediction. So the outline for the talk is just to set the scene, a small discussion on selective breeding, then a description of what genomic prediction actually is, and then launching into different machine learning methods for genomic prediction. So selective breeding is a powerful science. It's resulted in very efficient crop and livestock production to produce the food that we consume. So the idea is behind selective breeding that you select the parents with the highest additive genetic merit to breed the next generation. So you move the mean of the population generation by generation for the traits you're interested in. To do this, we estimate breeding values, the genetic value, additive genetic value of each animal that is a potential to be a parent of the next generation, usually using best linear unbiased prediction. So best linear unbiased prediction, we'll go into more detail on what this is, but in this case it takes into account trait records on individuals as well as pedigree, for example. And what we're doing here with the best linear unbiased prediction is disentangling in the phenotype the genetic effects from the environmental effects. So we get an estimate of just the genetic effects that are going to be passed on to the next generation. These estimated breeding values and the theory behind them is very much based on the infinitesimal model, which was developed by Fisher in 1918. And this model says that an infinite number of genes of infinitely small effect underpin the variation that we observe in the traits that we want to breed for. As I said, this selective breeding technology is a very powerful science. You can see that in the species where selection has taken place. For example, in chickens, the time to market weight for meat chickens has decreased from 16 to 5 weeks in 30 years. For Atlantic salmon, in just 7 generations, the feed conversion efficiency has improved by 140%. In crops, an example here is for corn, maize, where the selective breeding programs have resulted in an increase in yield of 77 kilograms per hectare per year. So that's quite impressive. So that's been achieved using pedigree and trait record information to derive our estimated breeding values, the genetic values of the animals. In the genomic era, of course, we have additional information, which is information about the genomes of our animals, variation in the genomes of our animals and crops. And the most abundant form of variation at the genome level are SNPs, single nucleotide polymorphisms, which are differences in the genome sequence between animals. And these are very abundant. For example, in the worldwide cattle population, there's probably 100 million SNPs in the population, the global population. Acquiring this SNP information has become increasingly cheap and straightforward with the advent of technologies like SNP arrays, for example, where animals or individual plants can be genotyped for 50,000, 100,000 SNPs at once using these array technologies. And of course, today, low-fold sequence coverage is also used to derive this genomic information. So we can acquire this genomic information, these SNP genotypes, whether an individual's A or T, or AT or AA or TT at an individual SNP for many, many SNPs across the genome. The question is, how do we translate this into estimated breeding values? How do we use this information? So this is what I'm about to explain is genomic prediction. So the first thing you need is a reference population where you have individuals measured for the trait of interest. And you need a large number of individuals because typically the effects of the genes or mutations you're trying to track are quite small. And you also need genotypes on these individuals. For example, 50,000 SNPs or 100,000 SNPs across the genome. This is just a cartoon of what that information looks like. So the red line is a chromosome, where you have an M is a marker, say a SNP, and where you have a Q is an underlying mutation that actually affects the trait. So just to give you an example, at this particular SNP, the difference between the animals carrying the AA genotype and the TT genotype might be 0.04 grams of growth. So a very small difference, but when you add up over all the mutations across the genome and the SNP effects that capture those mutations, you can capture the vast majority of the variation due to those mutations. So each of these SNPs is going to carry a weight, which is the effect of the second allele on the trait. So in our previous example, A would be the first allele, T would be the second allele, and the effect would be 0.02. And you're going to have one weight for each of the SNPs. So if there's 50,000 SNPs, you're going to have 50,000 weights. And this is your prediction equation. So the prediction equation is just a vector of 50,000 weights, one for each SNP. Once you have that, for breeding, you're really in business because you can go into the population, your candidates, potential parents for the next generation, genotype them, run their genotypes through the prediction equation, get their estimated breeding values, and then select them. And today we're going to focus on different methods for getting this prediction equation and producing genomic breeding values. So the challenge here is often, very often, you've got more parameters than records. So you have a P much greater than N problem. So for example, you might have 50,000 SNPs, but you've only got 5,000 lines measured for a trait, and you've got to estimate all those parameters. So we need methods that can deal with this. Perhaps the most widely used method is BLUP, Best Linear Unbiased Prediction, and its equivalent GBLUP. This method assumes many mutations of small effect, very close to the infinitesimal model, in fact. So to get your BLUP estimates of your genetic effects, you need to fit a linear model to your data. And this is a simple linear model where Y would be your phenotypes, your trait records, mu is the mean, X is a matrix of the genotypes for each individual for all the SNPs. So X has one row per individual and number of markers columns. The betas are the random effects of the SNPs, and E is a vector of random residuals. And the really key point with the BLUP method and its equivalent GBLUP, which actually works on genomic relationships, they both assume the SNP effects come from a normal distribution with the same variance. So the betas are distributed as a normal with zero variance and the SNP variance. So this is just an illustration, and this, assuming this distribution, means all the SNPs have an effect and they all have very small effects. When you invoke this assumption in your linear model and solve it with the mixed model equations, what it actually does, invoking this assumption, is it results in shrinkage of your SNP effects towards the mean, and the degree of shrinkage is proportional to the amount of data for the effect. So the less data you have for a SNP, the more the effect will be shrunk towards zero. You may have heard of Bayesian models for genomic prediction. These models assume or make different assumptions about the distribution of SNP effects. So for BLUP, as we said, assumes that every SNP has a small effect, and these effects follow a normal. Bayes-A assumes that some SNP have a very small effect, but it allows some moderate to large effects. And it does this by using a student's t distribution as the prior. In some cases, for example, when you've got a very large number of SNP, you may want to assume that some of them have a zero effect. And Bayes-B allows you to do this. So it has a prior which has a large spike at zero and then a t distribution. So many SNPs with a zero effect and some with a moderate to large effect. Bayes-R is a little bit more flexible again. It assumes a proportion of SNP have a zero effect, some SNP have a small effect, and some have a moderate to large effect. So it's actually a multivariate normal prior. So visually, again, BLUP assumes this single normal distribution. So all SNPs have a small effect. Bayes-R has this spike at zero, and then it has multiple distributions allowing larger and larger effects. So the degree of shrinkage is actually variable in Bayes-R, and you'll have less shrinkage of moderate to large effects if the data support that. Okay, just to show you the benefits of these two models, this is an experiment with about 800,000 SNPs. The training set was 3,000 bulls, 8,500 cows, all with production records. And there was a validation set of 262 bulls for the Holstein breed, 105 for the Jersey breed, and 114 for the Australian Red Bulls. So what we did was derive the SNP effects in the training set, then use those SNP effects to predict GBV for the validation bulls, and then we correlated those genomic breeding values with the performance of the bulls, which was actually based on the average of their daughters' performance for milk production. And this was the results. So there's a range of traits here. The one I'd like to focus on is fat percent. So for this trait, there is actually a mutation of moderate to large effect. And what you'll observe is the Bayes-R actually has a higher accuracy than the BLUP for this. And that's because the Bayes-R is shrinking that large effect less than what happens in the BLUP. So the accuracy is actually higher from the Bayes-R in this case. Okay, and this just shows why there's an advantage to Bayes-R. It's actually putting all its weight on a single SNP effect that you can see with the large effect on the y-axis compared to BLUP. Because it assumes all the effects are so small, it can't put all the weight on the SNP that's very close to the mutation. It actually has to smear it across multiple SNPs. Okay, so those methods, BLUP and Bayes-R and the other Bayesian methods, have been used very widely in genomic prediction. So what about machine learning for genomic prediction? So many machine learning approaches have been tried for genomic prediction. Here's just a snapshot: random forest, multi-layer perceptron, convoluted neural nets, attention networks, and hybrid schemes. And we'll look at the performance of each of those. So the data set we're going to look at to evaluate the performance of these methods is a sugarcane data set. And all this work was done by a PhD student of mine, Chen-Song Chen, who did a terrific job. So why sugarcane? It's an interesting candidate for assessing these machine learning methods because it has a highly complex genome. It's actually the hybrid of two species, and it's anything from octa to decaploid, so a very complex genome. It also has a lot of non-additive genetic variation for key traits. So TCH is total cane harvested, and VAA is the epistatic variation for this trait, and you can see it's quite large there with the green bar. It's also clonally propagated, which is good because it means you can take advantage of the fact that a clone has good both additive variation or additive effect and non-additive effects to get a great total clonal effect. Okay, so the models that Chen-Song tested in the data set included the convolutional neural network, multi-layer perceptron, and random forest. And I think you've learnt about these methods during the course. The actual data set, there were almost 4,000 clones, genotyped for 26,000 SNPs across multiple regions and years. And what's been done here is we've used all years except the final year as the training set for these methods, and then predicted the performance of the final year and correlated the prediction with the actual performance. So that's the correlations that you'll see. So the results for the different methods. So A here is a straight BLUP with additive effects. AD includes dominance, and ADE includes epistatic effects in the BLUP. RF is random forest. You've got numeric and binary convoluted neural networks, depending on how you code the data, and you've got the multi-layer perceptron. The y-axis is that correlation between predicted performance and actual performance. What you can see is the blup does very well across total cane harvested, commercial cane sugar and fibre. The machine learning methods, particularly random forest, do almost as well, but not quite as well. So close, almost competitive. Just to look at another data set to evaluate these methods. This is for disease of sugarcane. Smut and pachymetra are two disease traits. We had nearly 5,000 clones assessed for this smut, a fungal disease, and pachymetra, nearly 2,000 clones. And they were scored for the severity of disease. And these two populations were split into reference and validation. This time, Chensong has used an ML method using the attention network. And the attention network underpins the generative pre-trained transformers, or the GPT methods that are so widely used today. So here's the prediction accuracies for SMUT. Again, accuracy on the y-axis. You can see the attention network and two Bayesian methods. In this case, the attention network is actually slightly better than BLUP, which is the A there, but not quite as good as the Bayesian methods. An interesting question is, why does BLUP do so well? It seems like a somewhat naive model that all the SNPs have a small effect across the whole genome. So let's look into some evidence about the architecture of traits that we're interested in. And I'll use the example here of a classic complex trait, which is human height. So in this study, to look at the architecture of height, genetic architecture, 5.4 million people were measured and genotyped. So a huge data set. When that data set was analysed, there were 12,000 independent variants affecting the trait, and the largest of these explain less than 1% of the variation. So that's not infinitesimal, but it's quite close. Very large number of effects, all with a small size. So all models are wrong, but this BLUP assumption might be reasonably close to reality, which might explain why the BLUP method does so well. So if that's true, what's next for our machine learning methods? One idea is to start to combine the best features of the different methods. So Chensong looked at this in a very simple approach to hybridizing the methods, and he used methods like Bayes-R to identify a subset of SNPs that should go into the attention network prediction method. So what he does is he has all 26,000 markers. He cuts them down to a subset that have a high probability of having an effect with Bayes-R, and then includes just those methods in the prediction, the attention network. And you can see, again, A is the BLUP model. You add in dominance, epistasis. We have our Bayes-R model with all the SNPs. But then the hybrid with the Bayes-R PIP is in the purple, It's just taking the subset of markers identified by a Bayes R. And you can see it gets a slightly higher accuracy than the other methods. And this is true for both SMUT and Pachymetra. So this hybrid model is giving slightly higher accuracies. And it's actually true whether you use Bayes R to pick your SNPs or a GUR to pick your subset. So this is one promising area. And other people, other groups have used other approaches to combine the best features of each method. So Mark Cooper's group has been looking at these ensemble methods that combine the features of multiple methods. So this paper by Tamura looks at a couple of traits and looks at the accuracy of predictions from the different methods, and then finally a method that combines the information from all the methods, the ensemble method. And you can see this method, ensemble method, does a little bit better than all the other methods. Okay, another prospective area for machine learning and genomic prediction, I think, is to add information from additional types of data. For example, omics data. So epigenomics, transcriptomics, proteomics. And a paper by Zhao et al. Proposed a way to do this by considering these different omics features as different layers in their neural network. And they showed a benefit from doing that. You could also think about including genome annotations, whether a SNP is in a coding region or non-coding, controlling gene expression, regulatory region, and so on. So that brings me to the conclusion. Really, to date, just using straight machine learning methods haven't showed a great advantage over our BLUT methods. However, I think in the future, hybrid and ensemble methods that take advantage of the best features of each method have potential. Certainly adding different layers corresponding to omics and annotations could improve predictions. Also using multi-trait, multi-environment information may be useful in these ML methods. And finally, I think it's worth considering that we need very large data sets to really get the advantage out of these ML methods. They have millions, often millions of parameters. To take advantage of that, we need very large data sets. So international collaboration to build these data sets is a good way forward.